for joining in. You're joining the session workshop for the top 3 abstracts in the path to a cure. It is really my pleasure to chair this session. My name is Alejandro Pezzulo and I'm at the University of Iowa. And I'm co-chairing this session with Dr. Calvin Cotton, who is at the senior researcher at the CF Foundation. He's going to be working mostly behind the scenes, coordinating all of the questions. And I wanted to start by introducing our poster presenters. Okay. First, we're going to have Dr. Feng Liang, who is a senior scientist and a team leader at the CFTR lab in Lexington, Massachusetts. Second, we're going to have Dr. Melissa Calton, who is Associate Director of Translational Medicine at 4D Molecular Therapeutics in Emeryville, California. And third, we're going to have Anya Joint, who is a PhD graduate student in the lab of Dr. Gary Cutting at Johns Hopkins University in Baltimore. I wanted to remind everyone to please submit your questions using the Q&A tab, and we'll make sure to address them appropriately. We'll do 10-minute presentations, followed each by 5 minutes of discussion, and we'll ask all of the questions at the end of the 10-minute presentation by each presenter. Okay? So, without further ado, I want to welcome our first presenter, Dr. Feng Liang.
Good afternoon. I appreciate the organizer giving me the opportunity to present our work on targeting translation termination for pharmacological read-through modulation of CFTR PTC variants. The work is a collaboration of CFTR lab with Van Drake Research and the University of Alabama at Birmingham. The credit goes to all the folks that are working on the bench and those folks that are supporting us. No disclosure here. The only interest here is the drug. First, the background of PTC, premature termination codon in abbreviation PTC, also known as nonsense mutations, affect about 10% of people with cystic fibrosis. So far, there's no modulator therapy for this class. Therefore, premature termination is one major challenge on path to cure of cystic fibrosis. And so PTC simply means a termination codon before the natural stop codon of the gene. And we view this largely as a translation problem. And when ribosome scanning through the mRNA, and in this case, CFTR encounter PTC, and that will trigger the termination process. In eukaryotic, that's initiated by eukaryotic release factor 1 and 3 forming tertiary complex coming to the ribosome A site and to stimulate the termination process. In order for read-through to happen, that means elongation, and we need to have a near cognate suppressor tRNA to come into these locations and to compete with the termination process and get the translation going. We largely view this as a competitive process. Well, we think if we can attenuate the termination process, and we may have a little bit higher chance to have the near cognate amino acid moving to the elongation that is read-through. So this is why ERF1 and ERF3A can be a target for pharmacological read-through of PTC modulation. Our approach is a virtual screen approach. We start with the in silico docking of small molecules into X-ray structures of ERF1 and ERF3A. And once identified biological molecules, and we can plug in that into biological screen and follow up with SAR analysis. We initiate this effort in the reporter-based PTC read-through assay. Once accumulated compound with certain level of efficacy, we move that into CFTR-related model system that is native CFTR PTC to ask the question whether it can work for the patient. Showing here is our virtual screening process. The CFFT compound collection coming majority with the general chemistry, covering all the spaces there, and with a small focus on the small libraries there. And then we dock the CFFDI set into EIF1 X-ray structure and the EIF3A X-ray structure. We selected top scoring compound. Fortunately, majority of the top scoring compound are still available in the lab. We can plug in that into biological assays. The model system we initiate is a model developed by Dave Bedwell's lab in University of Birmingham. It's FRT cell-based nanolook assay where the X mutation is in the open reading frame of the nanolook. And this model also incorporates the beta globin mini gene into the frame expression cassette, so that to capture nonsense mediated mRNA decay. And in the screen, we use a low concentration of G401A to stimulate the assay. And then on top of that, we test all the compound in 8-point dose response, scanning 2 log range from 0.2 to 30 micromole range. The summarized here is the Emax of the dose response. And the x-axis is in log phase and of this waterfall representation of the compound. Bottom line is that we have hits coming out. To summarize the compound process here is that we start with 720 virtual compound, moving to 32 hits that fall into 5-hit scaffold. Then, given we don't have red chemistry in the lab, we just cataloging the compound through SCR, looking for structure analog. From public space, we identify 271 analog. We plug into SCR analogs into biological test. The model system we utilize here is very similar to the initial models and just switching the cell line into 16 HBE 14O- cells. Still have X mutations in the nanolook and with beta globin mini intron to capture NMD. And in the assay, we also use G401A to sensitize the assay and followed by compound testing in 8-point dose response. Shown here is a result in linear phase and of the Emax. And as you can see, only a small proportion of compound with a pretty reasonable efficacy, and some more compound has a relatively weaker efficacy. But this is enough for us to start the validation process. For the validation process, we ask the question whether the compound will work for CFTR. And so we use natural PTC models, and that is REM62X. And the assay we utilize here is the full-length CFTR ELISA, which is full-length CFTR ELISA, and which was developed with the help of the community. And in the full-length CFTR ELISA, we utilize 2 antibodies, either at the C terminal of the CFTR or the near C terminal. So the design like this is a means to capture full length rather than any kind of a truncated version, in this case, 1161. Showing here is the baseline. This is basically scientific 0 in the ELISA assay. And as you can see, with low concentration G401A, that's either minimum or borderline or no activity at all. On top of that, if we apply the compound with the dose response, in this case is 245 with 2 different doses, we observe a significant dose response, and which kind of saturate at about 5% of a wild type level of protein. To put that in perspective, and the VXA09 effect for phi-phi-del is in this assay is also 5%. So this activity is pretty reasonable for the first pass analysis. The other part is that the sensitivity of this assay is about 1% of the wild type. With 5%, we can have the confidence to make the validation call to say this compound work for read-through. Encouraged by this assay, we asked the other question about NMD. And as you may be aware, nonsense-mediated mRNA decay is a process that happens in lots of PTC mutations, and they simply RNA surveillance mechanism reduce the level of the PTC message. for most of the CFTR mutations, roughly will have 20 to 30% wild type level of message on the mutation, which is also true for R1162X. So in CFFT lab, we follow the vision of Dave Bedwell and Margarita Amaral, in that if we taking care of the primary defect of the read-through, the NMD will be resolved as a consequence. We put that hypothesis into a test for this 245 compound. What we observe here is that this baseline of 20 to 30% wild type level of expression, and with the compound application, significant increase to about threefold of the baseline with a solid dose response. And so at this point, we look at this compound has multiple profiles, which we are pretty interested. One is that it has a biological read-through, and two, it reverses NMD. So this is the criteria we use for the compound testing. And to summarize the validation part, this is still an ongoing process. We validate multiple scaffold, multiple compound, but I'm not ready to give a summary of the validation, but the result is pretty efficient. With that, I'm moving to a summary. In silico docking of that is a virtual screen of the in-house library into two X-ray structures of ERF1 and the ERF3A lead to the identification about the 720 compound, which we plug into biological test. We start with a reporter-based model system in the FRT cells with a PTC model in Nanolook, and then we identify 32 hits and falling into 5 hit scaffold. Follow up with the cataloging SER, we identified 271 structure analog and confirm in the FCR analysis, starting from Nanolook, moving into native CFTR PTC models. In conclusion, our study lead to the discovery of 5 hit scaffold with efficient read-through in native models of CFTR PTC variants. Thank you so much for your attention, and so I'm open for questions and discussions. Thank you so much.
Okay, thank you so much, Dr. Liang. So we are receiving some questions right now. So I'm going to start with the first one. So are FRT cells reliable predictors of PTC read-through in other cell types?
The answer is I don't know, and the majority of people believe it's not. And there are a lot. The issue behind is more or less is different models may have different threshold. My current view at this point is that PTC is a lot more tolerant than most other systems. And for example, compared to 16HB and the primary cells. So this cell type specific phenomena may be just quantitative or maybe qualitative, but yet to resolve. But a quick answer is that FRT is not necessarily the best model moving into questions about the primary cells.
Okay, thank you. Another question is, what counter screens do you plan to use to track on-target read-through on other genes?
We, as counter screens, so the one concern is about the cytotoxicity. There are lots of defects over there, and we definitely need to address those questions there because we are touching essential cellular models. And cytotoxicity is one thing, and translation fidelity is another thing, and probably there will be more. I think in the end, we probably have one efficacy screen, really quite a lot of counter screen and capture different characteristics we don't want to have within a compound.
Okay. Thank you very much, Dr. Liang. So we're going to move on to our second presentation by Dr. Melissa Calton from 4D Molecular Therapeutics. Please go ahead.
Hi, sorry about that. Thank you to the organizers for selecting our abstract and for the opportunity and privilege of speaking at today's workshop. I'm presenting the work from our current work from the abstract of number 514. A101 demonstrates, identified by directed evolution in non-human primates, demonstrates robust gene delivery both in vitro and in vivo. And I'd like to just point out that this is a work of predominantly the majority of the 4DMT family. My disclosure is that I am a full-time employee of 4D Molecular Therapeutics. So really, gene therapy for cystic fibrosis would hold significant promise if adeno-associated virus or AAV vectors could be engineered for more efficient and specific transduction of lung tissue following aerosol delivery. Therapeutic vector evolution, it's a directed evolution approach, employs non-human primates to invent novel AAV vectors with enhanced tropism for target tissues, and in for cystic fibrosis, that would be the lung, when delivered by clinically relevant routes of administration. So really our goal is to discover and characterize a vector capable of efficient and preferential gene delivery to the lung epithelia and to overcome the delivery hurdle of AAV to really deliver our payload or cargo in the primate lung. And so as a brief description of our therapeutic vector evolution platform is that a viral library is created by mutating the cap chain of naturally occurring AAV serotypes. These different mutations can include peptide insertions, they can be shuffles to create chimeras, they can be point mutations that are either random or by rational design, and a variety of other ways in which you can create, and what we've created is 1 billion vector variants in our libraries. Each virus is then packaged and purified, where each virus is composed of a mutant capsid surrounding the capsid, which encodes that capsid. Libraries are then placed under a selective pressure, and that would depend on what your target vector profile is for your specific indication or target tissue. So for example, you could incubate with human antibodies, you can deliver to ex vivo models, you can deliver to non-human primates, and you can use a clinically relevant way to deliver those libraries. Successful viruses are amplified and recovered and then enriched through repeated selection. So you keep doing this until you identify the number of hits or the viral DNA that has the vector profile that you are looking for. And this is what we would call a 4DMT lead variant. And so for the sake of the cystic fibrosis program at 4D, we use therapeutic vector evolution in the non-human primate lung. We believe that that non-human primate is the most relevant species. Prior to administration, all of the libraries were incubated with human IVIG, and then they were dosed to the non-human primate using a clinical device by aerosolization. We use the AeroEclipse 2 breath-actuated nebulizer, which is clinically available. It's fitted with an endotrach tube, and it was delivered to the monkeys that were anesthetized and placed in an upright seated position. So we started with 1 billion variants, and through using directed evolution and our therapeutic vector evolution platform, we got our lead vector, which is A101, which we believe has enhanced transduction, enhanced antibody resistance, and enhanced delivery. And again, this vector, what we're calling is A101. A101 is resistant to human anti-AAV antibodies. What you're seeing here is percent transduction of HEC2V6 cells in the presence of different concentrations of IVIG. And you can see here that in the dark blue, A101 has a greater than 4-fold improvement compared to AAV5, and even more so when you compare to AAV1, AAV2, AAV8, and AAV9. Moving on, we have our vector delivering the reporter gene payload, EGFP, driven by the CAG promoter, and looked at its transduction ability in human cells. We evaluated the transduction efficiency in 16 HBA cells and in non-CF, so healthy ex vivo ALI cultures. In the 16 HBA, you can see really robust delivery and expression of our EGFP. And then in the ALI cultures, you see a real true dose response as we increase the MOI. What we next wanted to do is really characterize this the ability of this vector to carry the cargo, this reporter gene in vivo. So we used non-human primates. We dosed 3 of them with approximately 1E13 VGs total, again using this AeroEclipse nebulizer. The in-life is 8 weeks. We collected the lung, and you can see here on this far right image that the left lung was collected for a variety of areas throughout for H&E for lung for histopathology and immunofluorescence staining. And then on the right, we collected a variety of different areas for qPCR to look at viral genome localization, distribution, and ELISA for the payload protein expression. We also collected heart, liver, skeletal muscle, kidney, spleen, and brain and spinal cord to really look at the non-lung systemic distribution. We also did histopathology and serum and collected serum for hematology and clinical chemistry to look at any initial adverse safety findings. And really what we found was that A101 delivers payload and expresses the GFP protein to the lungs. It is robust delivery and with minimal systemic distribution. So on the left, you see the qPCR from looking at viral genomes, so viral genome copies per microgram of DNA. You can see that the lung, we have about 10 to the 4 to 10 to the 5 copies, and this delivery was consistent between the animals. We do have detectable viral genomes in the liver, albeit at a much lower level, and that there was some detectable in the heart. None was detectable in the other organs looked at. All of the lung samples that we tested, so 16 from each animal, all had viral genomes present. When we look now to GFP protein expression by ELISA, all of the lung samples had detectable GFP expression. Some of the liver samples also had expression, albeit at a lower level, as was consistent with the viral genomes that we found. We found no GFP protein expression in the heart. So to look across now at immunofluorescence images to look at the protein expression distributed across the lung regions, here you're looking at representative images from the trachea, the bronchi, and the alveoli. You can see, so that in these images, blue is DAPI and the red color is staining for GFP. And what you can see is that in the trachea and bronchi, you can see that the epithelial layers here are stained. And we can see robust staining in the alveolar region as well. So really our aerosolized A101 vector reaches the bronchus and alveolar regions and expresses our payload or cargo, which is eGFP, the reporter gene. Looking at histopathology and hematology and clinical chemistry, there was no test article related adverse findings found. So kind of moving on from delivering this reporter gene payload of eGFP, we have now moved on to our product, our therapeutic gene product, which is 4D710. And this is delivering the codon optimized CFTR delta R therapeutic payload driven by the CMV173 promoter. And looking at the transduction efficiency and potency in human cells, using this HECTV6 cells, you can see that as increasing MOI, when we stain for CFTR antibody, you can see that we see increasing expression. Similar is seen, and that's by Western blot. Similar is seen looking at immunocytochemistry. You can see a nice dose response here, looking at the pink reddish cells compared to the and blue being DAPI. And then here at this higher magnification, you can see that staining for F-actin with the is shown in green. We are seeing cytoplasmic and cell membrane protein localization when we deliver this 4710 transduced in HECTV6 cells. We also looked at the 16 HBE cells, excuse me, and as we add increasing concentrations or MOI of our 4D710 gene product, when we probe for our transgene, you can see a nice dose response in mRNA expression. Further, when we look at, again, these non-CF, so healthy ALI cultures, these cultures have endogenous CFTR, so you can see that we can detect that in the black bar. When we add increasing MOIs of our 4710 product, excuse me, and use primers and probes specific for CFTR delta R, our therapeutic payload, that you can see in the non-transduced, we don't detect anything. At a 50K MOI, we see about 80% of endogenous, and then at 100,000, we see equal to or even greater than endogenous. And so put together all three of these different human cell types, we're seeing dose-dependent CFTR protein expression, localized to the membrane, and a dose-dependent mRNA expression in the human bronchial epithelial cells and ex vivo ALI cultures. And so in conclusion, we invented a novel AAV vector termed A101 by using therapeutic vector evolution. This A101 vector is resistant to human antibodies. A101 and the CF gene therapy product 4710 demonstrate efficient transduction of human cells in ex vivo lung cultures. A101 delivered by aerosol delivery is capable of safe, robust, and widespread transduction and transgene expression throughout the primate lung with really quite minimal systemic exposure. And so combined, we believe that this data represents a significant advance over existing AAV serotypes for the development of gene therapies for cystic fibrosis. And with that, I would just like to give our special thanks to the Cystic Fibrosis Foundation for their support and other members of the 4DMT process and analytical team and project management team. Thank you.
All right, thank you very much, Dr. Calton. So while we are waiting for some questions, I had a question. I wanted to start with that. So I was not familiar with using IVIG as a first step to select for viruses for which there are no antibodies in the population. Is there any chance that the lack of an antibody against a virus in the population correlates with the virus being just less efficient in general, and that's why it's not circulating and hasn't generated any antibodies?
Yeah, so that's a fantastic question. So there's kind of two parts to that. So you can imagine, we know that if you give something systemically, like through IV injection, that if there is some neutralizing antibodies there, that can decrease transduction efficiency. What is unclear is how this plays a role in the lung, whether serum neutralizing antibodies really will affect the transduction ability of an AAV virus. We just don't know yet. So yes, we employed this as a selective pressure, but really what that's going to amount to is still to be determined. Okay,
thank you. So another question from the audience is, were the HBE-ALI experiments done with apical or basolateral or bilateral exposures?
That's a great question. It was all done apically. We do our transduction on the apical side of those cultures.
Okay. And another question is, you mentioned that there are no human antibodies against your AAV. Have you looked to see if primates generate antibodies to it with repeat exposure?
I love this question. This is something I think about all the time, about really repeat dosing, to be honest, right? And so we have not looked yet. We have studies undergoing now to really evaluate this, to not only look at repeat dosing, but what if a human primate already has a really high level of serum level of antibodies, and does that affect our transduction? So I don't have an answer for you, but we are, I personally, and we are really interested in answering this.
Okay. Perfect. Thank you very much. So let's move on to our next presenter. So next is going to, Anya Joint is going to join us here. Anya Joint, soon to be doctor, is coming from Hopkins, and she's going to be talking about base editing for correcting splice site mutations in CFTR.
All right, great, thank you. So my name is Anya Joint. I'm a fifth-year graduate student in Gary Cutting's lab at Johns Hopkins, and I'm happy to be speaking to you today about work we've been doing with base editing for correction of canonical splice site variants of CFTR. I have nothing to disclose. So what is base editing? Single base editing is a newer flavor of CRISPR-Cas9 genome editing that has higher on-target efficiency and lower off-target efficiency. In this method, a single guide RNA brings a deaminase to a region of the genome, and then deamination will occur in a 5 to 6 nucleotide window. Then the endogenous cellular repair machinery is able to correct the deaminated bases, thus resulting in the desired transition, in this case from adenine to guanine. So for this project, we were focused on two canonical splice site variants. These were 273 + 1 G to A and 3718 - 1 G to A. And we chose these two variants because they were predicted to be good targets for base editing based on their sequence context. And while each of these variants is relatively rare, canonical splice site variants on the whole account for about 6.6% of all CFTR variants. And of the roughly 3,500 individuals with CF who harbor at least one canonical splice site variant, nearly 30% are currently ineligible for modulator therapies. Thus, this type of approach could benefit a large number of individuals. So to begin studying these variants, we introduced them into expression mini-genes, or EMGs, which are constructs containing all of the exons of CFTR and select full-length or abridged CFTR intronic sequences, thus allowing for study of both RNA and protein-level consequences. So to understand why these two variants and canonical splice site variants in general are good candidates for this type of approach, it's important to understand the molecular consequences of these variants. So 273 + 1 G to A impacts this donor splice site and results in complete skipping of exon 3, and this ultimately leads to generation of only shortened CFTR protein. Likewise, 3718 - 1 G to A results in complete missplicing, in this case through disruption of an acceptor splice site, with one misspliced isoform skipping a single nucleotide of exon 23, leading to a frame shift and ultimately decay of transcript, and a second misspliced isoform skipping exon 23 in its entirety, leading to an in-frame deletion and production of shortened protein. So notably, neither of these variants allow for production of any full-length CFTR protein, and thus these would not be expected to be very good candidates for protein-level therapies such as modulators. So to begin our preliminary work testing the efficacy of this treatment approach, we used HEC293 flip cells stably expressing our EMG constructs. And you can see in this Western blot that our wild-type EMG produces abundant, full-length, fully processed CFTR protein, or band C, while our delta F508 control here on the right produces only immature misfolded protein, or band B. So focusing in on these two lanes here, we found that introduction of the variant 3718 - 1 G to A resulted in production of only this shortened protein isoform, which was consistent with our RNA level result showing production of an in-frame deletion misspliced isoform. Notably, after a treatment with base editors, we do see restoration of some fraction of full-length CFTR protein. So given this result, we then wanted to know if we were recovering sufficient protein to have a functional consequence. So in order to be able to study CFTR channel function, we moved into a CF bronchial epithelial cell line previously developed in our lab called CF8s, which allows for stable expression of our expression mini-gene constructs. And so I want to take a moment here to emphasize that in all of our experiments, we are delivering our base editors via transfection without any selection for successfully transfected or edited cells, and then we propagate this pool of cells for downstream analysis. So in our edited cell line, only some fraction of the cells will have the desired correction. And we chose to take this approach because we feel that it's more representative of what would be occurring in the case of in vivo base editing. So here we are comparing mock transfected cells to cells transfected with base editing reagents, and using RT-PCR with a fluorescently tagged primer, we were able to quantify different size fragments corresponding to splice isoforms. So you can see here on the left that in mock transfected cells, there's only this one peak corresponding to the expected size of a splice isoform skipping exon 3. While after editing, we see the addition of this second smaller peak at the expected size of normally spliced transcript. And we can use the area under the curve of these peaks to estimate the relative ratios of these isoforms, and it comes out to about 10% of the total transcript in this cell line being normally spliced. Likewise, in CFBEs expressing 3718 - 1 G to A, we saw evidence of successful editing, in this case at the genomic DNA level. So here, looking at the Sanger sequencing chromatogram on the left, you can see that in mock transfected cells, we have only the original variant. Whereas on the right, after editing, in addition to the original variant, there's also this secondary peak corresponding to correction in some fraction of cells. Notably, we also see editing of this adjacent site, and this is a known limitation of base editing. Since the deamination step occurs in a window, you do run the risk of editing nearby adenines. However, one advantage of working with these canonical splice site variants is that these so-called bystander mutations are more likely to occur in introns, and they're unlikely to have any consequence greater than the original variant, since the original variant is already disrupting the most critical sequence in the splice site. So having seen evidence of editing in these cell lines, we then wanted to know if this was sufficient to produce a functional benefit. And so in order to test CFTR function, we use short circuit current measurements here with current on the y-axis and time on the x-axis. We used forskolin to increase cyclic AMP concentration and open channels, and the CFTR specific inhibitor INH172 to inhibit CFTR channel function. So you can see here that in mock transfected cells bearing 273+1 G to A, we see little to no CFTR channel function. However, after editing, we do see some recovery of function, and we can quantify this function by determining the portion of the increase in function that is inhibited by the CFTR specific inhibitor INH172, and this corresponds to the CFTR specific change in current. So similarly for 3718 minus 1 G to A, we see little to no function prior to editing, less than 1 microamp, and up to 15 microamps after editing. Lastly, we wanted to know if we could further augment the function restored by editing with addition of CFTR modulators. And so we treated cells overnight with either a DMSO vehicle control or the potentiator or the corrector compound lumicaftor, and then acutely with either DMSO or the potentiator compound ivacaftor. And so you can see here that for 273+1 G to A cells after editing, the function is further augmented by addition of modulators. And the same holds true for 3718 minus 1 G to A. So overall, we found that even with modest levels of editing, we were able to restore observable CFTR function, comparing the white bars here to the black bars, and this was augmented by addition of modulators. And while we were only able to achieve the clinically significant threshold of 10% wild type function in 3718 minus 1 G to A and with addition of modulators, we think that our editing efficiency can be further optimized by utilizing newer base editors as this technology continues to rapidly develop. In addition, methods such as RNA delivery or direct delivery of base editor proteins and alternative delivery strategies will hopefully allow us to achieve this desired threshold of 25% of wild type function. And so I'd like to take a moment to thank our collaborators in the Lu Lab for providing base editing reagents and expertise, and I'd be happy to take any questions.
Okay, thank you very much. That was a great presentation. So we have some questions from the audience. First question is, does bystander editing occur independent of target-based editing?
So if I understand what they're asking, if the, for example, if the other allele does not have my target variant, would that next door adenine still be edited? That I guess I would call more of an off target than a bystander, and that is a possibility. I haven't seen that in experiments where I have delivered the same guide RNA to wild type cells. I haven't seen any evidence of editing at either of the sites, but I don't think you could rule that out entirely.
Okay, thank you. So while we wait for more questions, I wanted to ask you, is base editing for splice site mutations when compared to base editing for other types of mutations, is it generally more or less efficient in comparison?
I I think it's kind of a difficult question to answer because the efficiency seems to be really dependent on the sequence context. And while there is some shared sequence in the splice site, because we're focused on these canonical splice site variants, the guide RNAs will extend outside of that like consensus region. So I think even between my two variants, I have seen differences in efficiency. So I'm not sure how it would compare to other sites.
Okay. And another question from the audience is, what is your hypothesis for why modulators would provide additional benefit to correctly edited cells? Is this related to modulator effect on wild-type CFTR, bystander mutations affecting CFTR function, or others?
Yeah, so our hypothesis is that the modulators are acting on the recovered wild-type CFTR, and we actually have a lot of evidence from studying other splice variants that allow for residual normal splicing and therefore some normal full-length wild-type CFTR to be produced, that modulators can act to augment the function of wild-type CFTR.
Okay, thank you. And let's wait a little bit for more questions. In the meantime, I wanted to ask you that, I guess this is a very common question for gene editing, is how will we get these reagents into the cell in vivo?
Yeah, that's kind of the big question that everyone is trying to answer. I think there are a lot of options that look promising, viral delivery methods, nanoparticles, but I think there's still a lot of work to be done to say what the winner will be.
Very good. Okay. Well, so with this, we're going to wrap up this session. I wanted to thank our co-chair, Dr. Cotton. I also wanted to thank the organizers of these workshops, the abstract reviewers, the CF Foundation, and most of all our presenters. Really beautiful work that was presented today. I think it really gets gets us excited about better therapies, and also the audience for these great questions. So thank you very much, everyone. Have a great night.